Welcome, my dear students. So today we are going to discuss about the lag operon. Is the word lag operon is frequently used in molecular biology. You have heard about it during your molecular biology classes, I think. But the lag operon, the term itself indicates lack one. What is this lack? Lack is nothing but lactose is the double sugar disaccharide lactose carbohydrate. Okay, so what does it mean operon and how it works, the things are going to discuss in this video lecture. So first of all, a few things you need to know: the control of gene expression, the lac gene expression. Each cell in the human contains all the genetic material for the growth and development of human. It's a very normal phenomena. Some of the genes will be need to expressed in all the time. These are the genes that are involved in of vital biochemical processes such as respiration. And other genes are not expressed all the time. They are switched on and off at need. So what does it mean? That means in the genome there are lots of genes. Genes are nothing but only the exons, not the intron. You are aware about that, I hope, because I have already discussed about intron and exon, junk DNA, satellite DNA in the classes. So that means certain genes can be expressed when genes are switched on. So here, when we are talking about the lag operon, so that means lactose operon genes actually they involved in biochemical processes. Biochemical processes are very much important, like respiration. Suppose you think without respiration it's very difficult to survive. Okay. So now the operon, the scientists Jacob Monod and Loaf, they won the Nobel Prize for the discovery of operon in prokaryotic cell. Prokaryotic mean unorganized unicellular organism like bacteria E. coli. Okay, they first identified the operon is a group of genes that are transcribed at the same times. So lac means the lactose. Operon means here you can understand. Operon means a group of genes. An operon is a group of genes that are transcribed at the same time. Okay, so they usually control an important biochemical process. Already we discussed like respiration and your more metabolism, digestion. Okay, so they are only found in prokaryotes. This is very important. If you want to search lag operon in eukaryotes, so you will fail. So lag operon means prokaryotes. Only in E. coli you can find. So now the lag operon consists of three genes as we discussed in the previous slide, each involved in processing the sugar lactose. That means the three genes, I will show you the three genes, they are involved in processing the synthesis of sugar lactose. Is the double sugar means the disaccharide. One of them is the gene for enzyme beta galactosidase. This enzyme hydrolyzes lactose into glucose and galactose. This is the main function. Okay, so you can write down in your class notebook what is the function of lag operon. So the function of lag operon is the processing of sugar lactose, and beta galactoside gene actually hydrolyze lactose and they produces glucose and galactose. This is the main function, and these are actually helpful for several biochemical processes to work out the biochemical processes. Now, adapting to the environment. As we are talking about, the lag operon is only found in eukaryotes. So here E. coli is the bacteria. Eukaryotes can use either glucose which is monosaccharide, or lactose, which is a disaccharide. However, lactose need to be hydrolyzed or digested fast. So the bacterium prefer to use glucose when it can. So that means lactose is hydrolyzed means digested. So that means to form the simplest form like the glucose monosaccharide so that easily it can be metabolized for the production of ATP in the bacterial cell. So now the four situation are possible. Here, first, the component of lac operon is the three genes ZYA, lac Z, lac Y, lac A. These are the called structural genes. What are the function? Is the lac Z is responsible for beta galactosidase synthesis enzyme. Lac Y is the permease. Lac A is the transacetylase. So what does it work? How does it work? Beta galactose hydrolyze lactose into glucose and galactose. This is the simplest form so that glucose is the monosaccharide can be digested or metabolized to for production of ATP. And permease is the lactose transporter into the cell. Permease, this gene helps the lactose transported into the cell. And this gene lac A adds acetyl group to the galactose. Okay. So here, this is the whole thing. Here you can see this is the gene structure. This is the structural gene, lac operon, and here is the operator gene. Operator gene, and here is the regulatory gene, lac L regulatory gene. That means it regulates the activity. Operator means it operates the activity of the all three genes. And this portion, including operator and the previous portion of regulatory gene, is called promoter, who promotes. So these are the term you need to remember in your mind. What is the lag operon? What is the structural genes? What are the promoter? What are the operator site or operator genes? What is the regulatory genes and how they work? These are the things you need to know. So now going to the previous slide. So fourth situation, actually we are talking about how lac operon works actually. So lac means lactose. So thing is that when glucose is present and lactose is absent, in the E. coli means the prokaryotes, E. coli does not produce beta galactosidase. So that means if glucose is present, lactose is absent. So there is no production of beta galactoside. In case of glucose is present, lactose is also present. Then only there is no production of beta galactosides because this presence of glucose it hampers the production of beta galactoside. And then glucose is absent. Okay. See, the glucose is not there in the E. coli cell, but the lactose is also absent. Then there is no production of beta galactosidase enzyme. But when glucose is absent, there is no glucose, but the lactose present. Means here glucose absent means no question of hampering or disturbing, but lactose is present. So lac, here is the lac. Lactose is present in the E. coli. Then only beta galactosidase enzyme will produce, which enzyme actually hydrolyze the lactose for production of glucose and galactose. Is understood? So now is the schematic diagram. Already we have discussed about it. The schematic lactose when it is absent, how it works. Lactose when it is absent is the lag operon gene, like ZYA gene is the operator side actually. The receptor protein here is the at the operator side. The receptor protein is continuously synthesized. It sites on the sequence of just DNA from the lag operon. It's called operator site. Is the operator gene and the operator site. Here is the seats and the repressor protein. This repressor protein blocks the promoter side here. Block the promoter side where RNA polymerase settle before the start transcribing. So it blocks the promoter side here because it is seated here. So that's why this promoter side cannot work. That's why these genes are not able to work. So no transcription will be there. If transcription will not be there, that means beta galactoside genes, this gene LACZ will not work. LACZ will not work. So lactose will not hydrolyze for production of glucose and galactose because the repressor protein is here and it blocks the activity of RNA polymerase. Next, when lactose is present, what it happens? A small amount of sugar alloctase is formed within the bacterial cell. This feeds into the repressor protein at the uh, another active site is the allosteric site here at the promoter site, and this causes the repressor protein to change its shape. See here, this allosteric site, this repressor protein, they actually allow to change its shape is called conformational change, and it can no longer sit on the operator site. RNA polymerase can now reach its promoter site because when they changes their conformational size, they just attached here. There is no question of attaching the RNA polymerase, so transcription can be started. So here you can see because these two proteins is attached here on the repressor protein, allosteric site, repressor protein. So there is no question of hampering or restriction to enter into the polymerase enzyme at the promoter region. So promoter side, the polymerase, DNA polymerase, sorry, RNA polymerase will come here and the transcription will start, means the LACZ activity will be started. So here, when both the glucose and lactose are present, so lactose, when glucose is present, it will hamper the activity of lactose. Okay. So see here, when glucose and lactose are present, RNA polymerase can sit on the promoter side, but it is unstable and keep falling off because when they sit here, they will fall it off. They will fall, certainly fall because there is no another, there is no another protein to keep it to attach the RNA polymerase for their activity. Okay, so that's why they can sit but unstable to keep falling off. So now when glucose is absent, see this is the fourth point. Glucose is absent means there is no question of, there is no question of restriction, means disturbance, because glucose is absent but lactose is present. So when lactose is present, the activator protein, actually another protein is needed for activator protein. This activator protein studies the RNA polymerase, studies the RNA polymerase. This stabilizes RNA polymerase. The activator protein only works when glucose is absent. Glucose is absent, only the activator protein is work. And activator protein, when it works, so RNA polymerase can bind to the activator protein. It will not fall it off, it will not fall, so it will binds with the activator protein, the RNA polymerase, and transcription will start. So once the transcription will start, that means the activity of LAC-Z for beta galactosides enzyme production will be started, and once it is started, so lactose will be hydrolyzed and produce glucose and galactose. So this is the overall situation, and is the summary, the four possible things. Glucose and lactose is present, no transcription. Glucose present, no lactose. So no question of transcription because lactose is not there. Here is also no glucose, no lactose, no transcription, but no glucose, only lactose. There is no hampering or disturbing elements, no glucose. So only transcription will occur. So I hope you understand this video. So nothing to worry. This is not so critical. Just watch it repeatedly, and I hope we can write if any question comes from this topic. Thank you. Thank you for watching.